I want to welcome everyone to MVZ Lunch. Uncharacteristically, I don't have any announcements this week. Does anybody have any announcements? Okay, then I will turn it over to Emily, who will introduce our speaker today.
Good afternoon, everyone. So it's my pleasure today to introduce Suzanne McGaugh. She's an assistant professor at the University of Minnesota. And Suzanne has really made some major contributions to our understanding of the genetic basis of adaptation using this really fascinating model system. Oh, can everyone hear me? Yeah, okay, good. Sorry, for a second I thought I was not talking to everyone. But she's looking at the genetic basis of adaptation using this really fascinating model system of Mexican cavefish that have independently undergone similar phenotypic, physiological, behavioral changes while adapting to these cave environments. And I hope we get to hear more about how she is shining a light on the evolution in the dark today. And addition to that, I also really admire the highly collaborative nature of her work within the cavefish system, but also across the broader genomics of adaptation community, and especially the effort that she's making in collaborating across departments at her university to better understand some of the inequities that are inherent in our traditional teaching strategies and STEM courses. And she's involved in designing some more inclusive teaching techniques, such as scientist spotlight assignments. And I encourage you all to go check it out. There's a paper on it that she co-authored on that technique in 2020, and it seems really fun. And a fun fact that I learned while researching Suzanne's accomplishments is that studying adaptation isn't the only thing she has in common with Darwin. She's also, too, has taken her own voyage of the beagle and is a very active in dog fostering. So check out her Twitter for some really cute photos of beagle puppies that are named after female scientists. And on that note, I can't really wait to hear what you have to say, Suzanne, and I'll turn it over to you.
Awesome, thanks. Wow, that was digging deep. Yeah, my husband Lex, he actually he shows up in Dan Schreider's like roadshow talk with a beagle foster that we've had too. So yeah, that's yeah, we do try to always name our beagle fosters after famous scientists. The last one was named Bates. Okay, so thanks so much for coming to my talk, and I'm really excited to share with you guys what my lab's been up to for the past few years. So it's really a cool time to be an evolutionary biologist. We have these classic model systems, and we understand a really good deal about the evolution and natural selection in these systems. We're also starting to understand the genetic basis of some of these textbook evolutionary examples. That's important for teaching us the paths that evolution uses. It's not so important just to like understand the genetic basis and the mutation that was made for that sake, right? How important is it to really know what exact mutation was done, right? But the thing is, once you start collecting all of those stories, you start to learn more on a whole picture about how evolution takes what mutations are there and shapes the natural world. And that's really where things get important and get cool. So for instance, whenever we think about the peppered moths, well, it's caused by a tandemly repeated transposable element that promotes cortex expression, which is a cell cycle gene. Whenever I first was giving this talk, I would always be like, and who would have guessed that a pigmentation mutation would be caused by a transposable element? And I was like, Barbara McClintock, that's who. So like now it seems so obvious, right? But then in addition, we know now that also a haplotype is sharing this ALX1 gene that's moving through different populations of Darwin's finches and integrating this little chunk of the genome that helps beaks get pointy. It's also the case that in stickleback fish, we have those pelvic spines that are caused by a loss of the enhancer. Well, turns out there is a fragile site in the DNA that is more likely to break during replication. And that is what is leading this to repeatedly cause loss of spines, is that fragile site part of it, the part of the genome that just seems to not do so well during replication. So we start to learn more and more about how genetics gets translated into phenotypic evolution whenever we collect these stories about the genetic basis of traits. And so we want to know in my lab, can we potentially predict the characteristics of adaptive substitutions? And now overall, my lab is guided by this goal. And we're just like at the very teeny tiny start of trying to figure this out. And so, you know, this is kind of great career path, because it's going to be forever that it's going to take us to do this. Okay, so to address this goal, we start to look at 3 different main themes in my lab. The first is repeated evolution, and the next is going to be genome content variation. We have a whole section of the lab that is collaborating with Candy Hirsch's lab on genome content variation, the pan genome in maize. Maize has massive amounts of genes that are different between different lines. And so we're looking at that to try to understand what this presence absence variation contributes as far as phenotypic evolution. And then we also look at pathway evolution, but today I'm really going to talk to you mainly about our repeated evolution stuff. So we use systems of repeated evolution to provide replication, like natural replication to evolutionary events, and to understand, you know, are we going to get same or similar genetic mechanisms being employed? Now, probably one of the most fascinating and amazing examples of this is in the tetrodotoxin bearing amphibians and the snakes that become resistant to them. So this is one of the most canonical and famous examples of convergent evolution that pops in my mind, at least. And you see the phylogeny of snakes, and each one of these colored things are snakes that have evolved resistance to tetrodotoxin bearing amphibians. And they eat these amphibians. Well, what's amazing here is that, you know, this is pretty strong selection pressure, because if you become paralyzed, you're going to die. But the thing is, they are doing it in the same way. So essentially, we see that it's not only the same gene, it's sodium channel 1.4, but it's also the same place in that same gene, right? And so this is really striking that they're not only using the same gene, they're using the same region. And it's even more striking when you step back and look at all of the vertebrate tree life and say, holy crap, pufferfish are doing the same thing. They're getting to tetrodotoxin resistance, also using the same protein and the same areas of the protein, right? So that's amazing whenever we really step back and say, okay, there's between 15,000 and 25,000 genes in most vertebrate species. They can be turned on and off in combinatorial ways, allowing just a multitude of options for creating phenotypes. Why does nature always seem to use this gene and this region of the gene? And that's especially important because when we start to look at mutational screens in the lab, you can see that you get phenocopies of the same or very similar phenotypes using lots of different mutations. So why are we only seeing a certain handful of those show up in nature? So this is what my lab is really interested in studying. And there's been some general themes that keep popping up in the literature. So the first is, okay, loci that are more important for phenotypic evolution, they could exhibit less deleterious pleiotropy. That's a totally feasible hypothesis for why we see these particular mutations contributing to phenotypic evolution more than you would expect just by random chance. It could be genomic context, like the fragile sites or the particular recombination rate area of the genome. And it's also potentially the length of the target size. So really long genes potentially get hit more by mutation, and that could cause them to lead to more phenotypic evolution. And that's kind of, you know, I'm going to use that as a segue to get us into Cavefish. And that's that OCA2 is probably our most famous gene in the Cavefish community. So OCA2 is a gene that causes albinism across a lot of different vertebrate species, and also a lot of invertebrate species too. It's a very, very long gene. It's 120 kb, and it is repeatedly causing albinism across many, many metazoans. In surface fish, we have this nice intact reading frame, but in Molino Cave, we have a deletion of exon 21, and then in a different origin of the cave phenotype of Osteonyx, which is the species of fish I work on, you have exon 24 that's deleted. And it seems like these deletions are very much driving albinism by themselves. They are necessary for causing albinism, and whenever you cross Molino and Pichon, they do not complement one another. So it seems like it's this gene and this gene only. So it could be that, you know, this is just a hypothesis, but OCA2 might be a nice example of a very long gene that repeatedly contributes to phenotypic evolution. So like Emily said in her really nice introduction, I study this cavefish system, and what I'm showing you here are the 5 main populations that we work on. So there's over 30 caves that have fish in them that have been discovered. And we usually use these 5 populations. You're going to see them pop up throughout the talk. So we have Molino, Pachon, Sabinos, Tanaha, and Chica. And hopefully I'll have time to touch on some brand new work we've just submitted on the Chica cave later in the talk. So it's important to note, and I'm going to show you the phylogeny that shows this, that there's 2 lineages of surface fish that gave rise each to at least 1 cave colonization. And so whenever we look at the behaviors that these fish have, I want to show you just a quick video because it's easier for me to show you that than to just talk at you with no visuals. So you can see these guys, they are getting around the tank really well, that they're completely blind. These are just pet ones. You can buy them at the pet store. Usually the ones that are in the pet store are from Chica Cave. They were collected like in the 1930s and have been propagated in labs, or well, sorry, in hobbyist tanks ever since. And so they have expanded number of neuromass that help them get around the tank. They also are really adept at figuring out when food gets around. So instead of feeding in the water column, they have this characteristic 45-degree angle that they have with the ground. So they have expanded taste buds all over their heads too. And so despite lacking eyes, they're able to basically function pretty normally. You can also note, even though these are aquarium fish, they're really, really fat. And in fact, whenever you see fish in the wild too, cavefish are usually fatter than surface fish are. So they lose weight a lot less quickly. They have more fat stores than surface fish. So if you're interested in these tribomorphic syndromes and you're really excited about them, you can read our book that was published in 2015. It's a little old now, but it will detail things on retinal degeneration, pigmentation reduction, neuromass cell expansions, the enhanced taste buds. They also have this weird vibration response behavior where if the cavefish detect a vibration in the water, they'll swim towards it because it's usually food, like it's usually bat droppings that's going into the water, or like a rogue spider that lost its way and fell in. And so they want to get onto that fast as possible. Whereas surface fish, if they detect a vibration in the water, that's usually a predator, so they don't swim towards it. So they've actually switched this behavior too. And then in addition, they have these interesting sleep phenotypes. I'll get into later in the talk, where they don't sleep as much as the surface fish do. And there's metabolic differences. We also have a cavefish genome that was published in 2014 with short reads, and then a full chromosome level assembly of the surface fish now that was published just about a month ago. And so we have both the cave and surface fish genomes to compare. And one really cool thing about this system is that you can interbreed the cave and surface fish and make F1s and then self those, get F, or not self, but interbreed those and get F2s. And then you're on your way to do Q-tail mapping for all of these different traits, which is incredibly useful. So today I'm going to talk to you about 2 main parts of the cavefish system we've been working on. The first is PopGen, and the second is going to be the evolution of sleep loss, but we'll get into that in a moment. So I'm going to just cover the basics with you that everybody asks whenever we first start talking about this system. So everybody wants to know, okay, are there actually independent origins of the cave phenotype? And do you know that? Have you plotted it on a tree? The answer is yes, and I'll show you some of that data in a second. Is there gene flow between the cave and surface or between caves? Yes, there absolutely is, but I will show you the data in a moment. How old are the cave populations? Not as old as we used to think. And the effective population sizes of the caves and surface are quite a bit larger than we thought too before we did this work. And then selection can most definitely drive the cave traits. And I'm going to show you all that data in just a moment. But this was all published in 2018. So again, this is a little bit of an older study now. And I'm just going to show this to you as like a get your feet wet into the system rather than throw you all the data that we have now. So we have Molino, Pachon, and Tanaha. Those are our 3 core caves we use for almost everything. Then we have Rio Choi to serve for surface population, and Rescombe. That's also a surface population. Now these 2 surface populations, even though they're close to one another, there's actually a giant waterfall. And so that has prevented the surface lineage that is Rio Choi from finding its way to mating with the surface lineage of Rascone. So these are the two surface lineages that I mentioned at the beginning of the talk that contributed to the two origins of cave. We also sequenced one of these innate, or two of these Aeneas samples, which served as an outgroup for all of our work. And we started this a long time ago, like 2013. And so we started with a pretty moderate number of samples and pretty low coverage, and just sequenced them with whole genome sequencing. And indeed, what we find is exactly what we were hoping to find, where we have, you know, polyphyletic cave morphs. So we have cave being sister taxa to the surface, and then here again we have a surface fish that's sister taxa to these caves. So we have these blue lineage, lineage 2 is what I might call that, and pink lineage, lineage 1. What's cool about this is that Tom Kono then took all of our data, ran it through DADDY, and then was able to estimate some divergence times. And so we have about, you know, 260,000 generations ago, we had the split of the two surface lineages. And then we had this time period about maybe 200,000 years ago, if you consider 1 year per one generation, where the fish were going into the caves. And it doesn't matter if you reported lineage 1 or lineage 2, just about the same time, both of those fish went into caves. And so we need to do a little bit more digging into our like paleo climate data to try to figure out what was going on in Mexico, in that region about 200,000 years ago, where those caves might have served as refugia. So that's great. We kind of know that these are pretty recent events of the origin of the cave phenotype. We also wanted to know just how much gene flow there was. And so this is an example of me and going into this cave where we often find surface fish getting washed in. And the surface fish, there's a contingent of cavefish biologists who say, okay, they die before they ever get to spawn. They get into the caves, they're so maladapted. They look like, you don't have to be a fish biologist to know that's not normal fish behavior. They don't, do that and live to tell about it too much longer. So it's thought that, most of these surface fish that get swept in become cavefish lunch. Well, we wanted to know if that was actually true or not. So again, Tom did some models with DADDY. And if you're need a refresher on how these DADDY models work, you have this ancestral population here represented by the A, and that can get split into 1 and then 2 in a very strict isolation model. That's what the SI is for. And then you can expand this so that you can have gene flow either during this separation, or maybe you have some ancestral migration and then a time period where it all stops, or maybe you had a time period where it was stopped and then it comes back together in a secondary contact model, meaning SC. So you can test all these models. You can also add another parameter in there that says, okay, we're going to allow for parts of the genome to flow pretty freely between these two populations. And then we're going to force some of the genome to be restricted in its gene flow, kind of mimicking, protected regions by selection. And sure enough, Tom found that for almost all of our population pairs, the secondary contact model, meaning that they were split for some amount of time, then come back together, and then parts of the genome are protected from gene flow, seems to fit our data the best. We can corroborate that with looking at, our standard admixture plots where we have each individual we sampled here as a bar. And what you can see, so this would be our blue lineage I was talking about earlier, and then our red pink lineage here, is that we can see transferring of the alleles from the surface into the cave of a different lineage. And then also you can see from this cave to this, this cave to the surface over here. So you see these relatively recent admixture events whenever you're starting to see like that amount, that maybe looks like about 25% of that individual from Rascone is coming from Tanaha. So that's pretty significant and relatively recent admixture event. We could take those things out that look like they are recent enough to have a big chunk part of them coming from another place, because we know those are recently admixed individuals, and we can say, what does the historical admixture look like? And sure enough, we also see evidence of historical admixture. We can estimate this and be able to say like, wow, so we used to think that the caves were all completely isolated from one another. Turns out the thickest arrows are representing something like 1 to 1.5 individuals per generation. This is a pretty far distance as well. So we're talking about, I don't know, maybe it's like about 3,000 kilometers, maybe not that far, but pretty far to be able to transfer those alleles. Now, it might not be exactly this cave transferring to this cave. It could be that, it's just that these alleles that are most similar. So there could be other caves in here that are actually contributing the alleles directly. But either way, we can basically do a take-home message here to say there's a decent amount of gene flow between the caves and surface. And sometimes we even see some cave alleles, find their way out into the surface populations too. Furthermore, we also see that the effective population sizes of the surface and the cave are a lot larger than we thought. So the surface fish are very similar to what we would see in zebrafish or in some species of Drosophila as far as their effective population size, and kind of give you an idea of what I'm talking about. Now, it used to be thought that, cave populations, the max out of all of the cave populations was 1,000 and as effective population size. And when we did the daddy modeling, and I'm referring to the Molino cave, which is the most inbred, or the most, I should say homozygous cave. It has an effective population size that's not so distant from humans. And then with Pachon and Tanaha, these other two caves, it's triple that essentially. So what we're learning here, if we take a broad step back and we say, okay, so these guys are a lot more recent than we thought, the cave populations. They're exchanging genes with the surface a lot more than we thought. And they have a higher effective population size. Well, that starts to change our thinking about how effective selection could be on cave-derived traits. Now, why that's important is because what's dominated a lot of thinking in cave fish biology is, you get into a cave, drift takes over, you don't use your eyes, and you lose them. And that's been the general theme, and it's been, that's from Darwin, but also it's prevalent in the literature not too long ago as well, that they are held up as the poster child of disuse and loss, right? Well, that just wasn't sitting right with us, knowing what we know now about the demographic parameters of the cave fish, and also that eyesight and image processing is energetically expensive. There might be selection to lose it, because especially in a nutrient low environment like a cave, you have to conserve the resources. So this has really important also implications. The demography really informed our expectations for how successful scans for selection might be, and scans for sweeps might be. So we collaborated with Reed Cartwright, who had recently put out a model. It was a 12 locus additive allele model that was saying, like, okay, we know there's about 12 loci or 12 QTL that have been mapped for eye size. That's not perfect, but starting point. Can we take our demographic estimates we've got from daddy, meaning the times of the split, the time of the coming back together, and the exchanging genes, and the effective population size, put all that into your model, and then start to see what the selection coefficient would need to be able to select for eyes. And so what I'm going to show you here is there's about 91,000 generations of isolation, followed by about 71,000 generations of them being connected again. And so, what you can see, and I'm just going to stop this for a second to orient you to this. So this is the selection coefficient. These would be things that have all cave alleles, right? It's all cavey. It's all coding for loss of eyes. And then the red would be very, very low cave alleles. So all the surface alleles, all the ancestral alleles. And so you'll start to see, like, around 91,000 generations, things start to bounce around a little bit, but what you're seeing is pretty quickly selection coefficients needed to increase cave alleles in the population don't need to be super crazy. So, we're seeing, even after migration starts rehappening again, we don't have to worry too much about needing that strong a selection. So these selection coefficients, so let's say we have 0.01 to 0.1, that range is what we're seeing would be needed to basically fix or almost nearly fix these cave alleles for blindness. They don't have to be all that high. So these are pretty standard selection coefficients in other selective sweeps and other wild populations. So this would be the selective sweep that, or selective coefficient that would be needed to bring pelvic spine reduction in freshwater populations of stickleback within about 8,000 generations. So it doesn't, so what our work showed is, hey, you know, this whole idea about drift and disuse maybe is a little outdated, and maybe we need to start thinking about these populations that selection could actually work on them. So this is also notable. This is the Molino cave, the one that's most homozygous, has the lowest effective population size. You would need even weaker selection coefficients in places like Tanaha or Pichon, where they have much higher effective population sizes. Okay, so why is this important, right? Well, part of it is that we are starting to dive into selective sweep scans using some methods by Dan Schreider that he's developed. And we started to look into these, like the most dumb way you can do this is say, let's look at the QTL and see what we see under the QTL. And what happens is that we started to look at this particular QTL that seems to have a lot of traits mapping to it. So it seems like many things that make a cavefish seem to be localized into this region on linkage group 2. And when we started to look at this region in depth, what we found is that, you know, between surfaces, like service population, the divergence was pretty much the same as genome wide. But when we looked at cave to surface, the divergence was higher. When we looked at cave to cave, especially the, you know, looking at all 3 caves we had, and remember those are 2 completely different origins, we saw very extreme low divergence. And what we think is happening is that gene flow may be transferring this little cave adapted chunk between the caves. So taking some of our newer data that we have now and trying to dig a little deeper into this linkage group 2, and I should mention there's another common co-mapping region here on linkage group 17, but we don't see these same patterns with that. It has a lot of traits that map to it, but we don't see these patterns of divergence, of super low divergence in the caves, like as if it's being transferred between caves. So this seems to be isolated to linkage group 2. And then Rachel Moran, who is absolutely one of the most phenomenal young scientists out there right now, I was lucky enough to have her join the lab, and she's starting to run these twist trees where it's basically getting these topologies, unrooted topologies across the genome. And this is that area that I was just showing you. And so you can start to see that there are some places where the genealogies just aren't, they're looking very much like, so this is Pichon and Molino. Those are 2 separate origins of cavefish. And those guys are, that particular region seems like it's grouping those together. So this is brand new. She just showed this to me last week. So we're still digging into this. It needs to be smoothed. We need to make some sense of it, but it does seem like something odd is going on there. We have 10x data from these populations, so we'll be certainly looking at whether there or not there's potential for inversions in that particular region too. Okay, so conclusions from this aspect of the talk is that definitely there's probably 2 lineages at least. There is gene flow. Cave populations are much younger. The effective population size is much higher. Selection can definitely drive these traits even when really polygenic. And one thing I wanted to mention, most of you guys know this, I'm absolutely sure. You know, but relative measures of divergence can be extremely misleading. And now I want to just take a quick aside as this being a poster child for this particular problem with FST. So when we measured our divergence estimates, we always measured them with DXY, and DXY is essentially pi. So this is meaning that you have like 2 chromosomes, and I have these little tick marks, right? So those represent SNPs. So I'm saying 7 differences, and let's say there was 100 possible sites. So that's how we just average. We just do average pairwise diversity. So that would be pi, right? Now, you can also do this between species or between populations and call it DXY, where you're essentially doing the exact same thing, right? So it has 10 differences. The yellow is supposed to represent a different population or a different species. And so you get a DXY the same way. So this is looking at all sites, invariant and variant sites. Now, there's an important component to FST, a pairwise FST, is that in order to have everybody on the equal playing field, you need to make sure that your pies are all pretty similar across populations, or else that component of the equation is going to be messing things up. And sure enough, that's what has happened in the cavefish community for the last several decades, because they all use FST. So when we're looking at pi and dxy, so that's what this is representing. Off diagonal is dxy, diagonal is going to be pi. So it's like, you know, the individuals to themselves essentially, and Choi is that surface population, and it's much more green, right? It's much more the very dissimilar. You can pick out any two Choi individuals, and they are actually going to be more different than if you picked out, let's say, a Pachon and a Tanaha individual. Like two random individuals from two different populations are going to actually be more similar in some cases than Choi, two individuals from Choi. It's that diverse. Whereas in Molino, this is just like this blood red that's showing you this thing is super homozygous. Everybody's basically the same. So that's a major violation of Pairwise Fst, and it plays out like this. So FST, we can think about it as dxy minus the average pairwise pi between the two populations. So like if I had two caves, I'm going to say like, what's my average cave pi between Molino and Pachon, right? And then, you know, over dxy. So what happens is that like when you decrease your pi, which happens with the caves because they have limited diversity, you're going to artificially pump up FST. And then when you compare like your FSTs between caves and between surfaces, you're not comparing apples to apples there. So this plays out like this. So I have average pi on the x-axis here. So this is, you know, for cave to cave, it's going to be really low. To surface, it's going to be really high. And whenever I do Molino to the other caves, their FST is screaming high. When I do surface to surface, well, their FST is really low. But that's all driven by the pi, because the surface to surface dxy is actually just a smidge higher than the cave to cave dxy. So for decades in the community, the mantra was caves are highly divergent from one another, surface fish are all panmictic. But what our data are showing, and I think I'm spending time on this because I think it's a nice example, that if you're reviewing a paper and someone's only using FST or whatever, you can say like, hey, you need to go see Herman et al. They show this, they have this laid out, that, you know, you're using pairwise FST when your pi's are all over the place. Please don't do this. Check out this crazy graph in Herman et al. So that's why I bring this up, because I think it's a nice example, is that, you know, you're really seeing a difference in the biology whenever you're looking at absolute versus relative measures. And they're telling, this is made us as a cavefish community go astray for quite a while. Because we know now that the DXY is actually, you know, surface isn't pigmented necessarily, and cavefish aren't that isolated. So what we've done so far, so I started out with this, but now we're definitely pumping up our sampling, and this is being led by Rachel Moran and Daniel Drabeck. And you know, with Rachel's new data, we're seeing basically the same thing that we've seen before. So this is just more cave and surface and population sampled, and the new and old lineage stuff is just the way we shorthand say lineage 1, lineage 2. And then we see that we probably have 3 origins of the cave phenotype. It could be this particular one might be caused by hybridization, that it's grouping with the surface populations out of like really significant hybridization. It actually probably needs to go here. So we need to figure that out. But for the most part, even with increased sampling, increased number of populations, we're still seeing just about 2, maybe 3, but I'm very suspect of that 3rd. So, okay, so I started out this talk and I said, hey, we really want to know in my lab, we want to look at independent origins of these phenotypes and be able to say, like, these are independent evolutionary events. What's going on in the genetics in these independent events? And then I told you a whole bunch of stories that rely on gene flow, right? So we really need to get populations that don't have this, that I can say, no, I know that these are de novo mutations. That's how I'm going to study this repeated evolution from the de novo mutations onward. And sure enough, I think we might have found a population like that, and that happens to be in Central Texas. So this is one of those serendipitous things where I was at a meeting and happened to run into Andy Glusenkamp, who is a Mexico-phile too, just loves to do field work in Mexico. Actually did his PhD in the same lab I did my undergrad in, so you never know how people in your past are going to show up later. He said, hey, I found some fish in a cave in Texas, and you should come check them out. And so the thing about these fish, they're still osteonyx, but osteonyx historical range until 1900 was the Rio Grande. Then some intrepid entrepreneurs decided to dump them into the rivers as bait fish. And so you can start to see this explosion of records by about 1950. And you can see our little cave population basically getting surrounded. Well, there were some surveys of this particular cave in the 1970s where they did not see any cavefish in the cave at all. That doesn't mean they weren't there, but as far as we know, they weren't in the cave in 1970. There was a major flood that happened in 1978. And after that, cavefish were seen in this particular cave. So we're only talking about 40 years that the cavefish have been in the cave. And with the help of several great people in my lab, we were able to collect some of these fish and bring them into the lab. So the cavefish have, and these are all done on wild caught fish, so there's that caveat of development affecting these phenotypes. So we're not going to call this evolution, we're just going to say that they're phenotypically different. And so cavefish are lighter than surface fish, and that's a significant difference. Cavefish have more neuromass around this particular bone. So this is the area where they're using to detect a lot of different vibrations for prey. And we have more of these little tiny neuromass. We're not looking at the canal neuromass, but the superficial ones than in the surface population. They also, if you starve them for 5 days, they act like what you would expect organisms that are adapted to low nutrient environments. They're like, when you finally let them eat, they're like, okay, well, there's food, whatever. But the surface fish are just on it. Like, I haven't eaten in 5 days. I desperately need this food. Cavefish just aren't that into it. Cavefish also seem to follow the walls. And so this seems to be a recurring phenotype across all different types of cavefish species, that they develop a wall following behavior. We also see that whenever we're looking here. So we put a fish in a bucket, we watch it for a day, and then quantify where it was. And the dark red areas are where it spent most of its time. That's basically tracing it. And so we see that, yeah, cavefish on the whole like to hang out by the walls. Surface fish don't really care all that much. And so, yes, this is all on phenotypic data on things that we collected in the wild, but we have now bred these fish, and the first batch have reached maturity, and we're going to be testing all these phenotypes again on lab-raised fish to see if this is genetic or not. If it turns out to be, then I really think that this population, and we've also discovered 2 additional populations since we started working down in Texas, that this population can really start to tell us a lot about the first steps in the evolutionary process of becoming troglodyte. And it could be really scientifically useful for a lot of different projects, as these Texas cave colonizations probably happen more than once. Okay, so now we're going to switch gears and start talking more about the sleep loss work. And I might have to go through this kind of quickly, but so here we need to talk about the definition of sleep first before we really dive in. A lot of times whenever I give this talk, people say, how do you know they're sleeping? They don't close their eyes, they have no eyes, and it's that we really rely on the behavioral definition of sleep. So a behavioral definition of sleep is that they have prolonged behavioral quiescence. This is often accompanied by a preferred location or characteristic posture. This means like you go to your bed and you lay down. You have a decreased responsiveness to your surroundings, so it takes longer for them to respond to a tapping or something like that. And then it can be reversible. This distinguishes sleep from being hibernation or coma. And then in addition, it's homeostatically regulated, meaning that if I keep them up, force them to not sleep, that they experience some rebound. And so this is the general 4 criteria that sleep researchers use to define sleep. And in fact, even the Cassiopeia, the upside down jellyfish, enters this period of nightly quiescence where their rhythmic pulsing slows and they exhibit these 4 criteria. That's absolutely outstanding. This means that sleep is, or at least behavioral quiescence, is ancient. It precedes the development of the central nervous system and even cephalization. So this seems to be an ancient phenotype. And in my opinion, it is not studied enough. And it's really cool because there's so much variation to study. So in mammals, we can see the big brown bat sleeps 20 hours a day, whereas an elephant sleeps, 2 hours per day. Elephants can go days without sleeping without any ill effects as far as we can tell. And it seems like one of the only correlates, like if we were to try to look at all metazoans and say, what is driving this difference? The only thing that can really be come up with, and it's not even that solid of a hypothesis though, is that carnivores sleep more than omnivores and they'll sleep more than herbivores. So if you have a really nutritionally dense diet, it leaves you more opportunity open to sleep. So that's what we think, right? And that seems to be the one generality we can make. Now that's going back to cavefish. This is really cool in that cavefish sleep less than surface fish. So this is, sleep bout duration. This is during the day. This is during the night. We have our 3 populations I've been talking to you about this entire talk. So Pichon and Merlino and Tanaha all sleep less. And that accumulates into a total sleep of about a quarter-ish as much as surface fish do. So what's amazing about this is that this is one of the only systems where these are evolved sleep differences. And so I can start to pick them apart using classical quantitative genetics. And we have a grant right now, and we're doing the mapping right now to try to understand the genetic basis through just classic mapping approaches. And it's also notable that humans, the sleep loss in humans correlates with increased appetite and insulin insensitivity, and short sleeping individuals are more likely to develop obesity and type 2 diabetes. Cavefish seem to mimic that as well. So they have lower sleep duration than surface fish. They eat a whole lot more than surface fish do. And then they also pack on the milligrams much faster than surface fish do. So their triglycerides are much higher. Cavefish are much fatter than surface fish do. So it's thought that maybe these are all adaptations to the cavefish being raised in a really nutrient poor environment. And so when food is readily available, they need to eat and eat and eat. And these metabolic changes have co-evolved to compensate for the limited or seasonal changes in food availability. And so testing that, the plasticity in sleep seems to be driven by nutrient availability. So if we starve surface fish for 30 days, they start to sleep less. So they start to shift their energy budget, or their time budget, to more towards finding food. And so they really start to sleep much less. The opposite happens with cavefish. If you starve cavefish, they're like, okay, I guess we're in the dry season and there's no food available, so I'm just going to sleep now. And so it's almost like we are putting them, by having these IACUC protocols that say you must feed your fish every single day, we're putting the cavefish in the state of constant sleep deprivation, because they are clicked into the wet season physiology of I need to eat and eat and eat, and I can't sleep. So we've been collaborating with Nick Roner from Stowers and 3 different labs from FAU on several different grants to figure out the genetic basis of the sleep phenotype, also figure out the genetic basis of the plasticity. And I'm just going to briefly touch on a couple things we've been working on, because I don't think I have too much more time. So we first wanted to know, okay, cavefish don't have as high baseline sleep as surface fish do, but do they still have the same sleep pressure as surface fish do? So your friend who can get by with 3 hours of sleep, do they still feel as sleepy as you do whenever they've missed that 3 hours of sleep? Do they still have that same rebound? And so we wanted to know is this homeostasis intact despite their need for less sleep? And in fact, it hasn't been studied really well about how this relationship is between baseline sleep duration and sleep pressure. That isn't really well explored. And so to do this, we took 3 different cave populations, sleep deprived them, and then flash froze them and did RNA-seq on them. And then we'd also do this with one of the surface populations. And to sleep deprive them throughout a 10-hour night, we had them on a shaker, and one time every minute, randomly, they would shake and not be able to fall asleep throughout the entire night. Now, this isn't the perfect proxy because it could also cause a little bit of damage to the fish and a lot of stress. And so we need to figure out a better way to keep them up, but it actually worked. And so this would be all through the day, the sleep deprived night, and then the recovery day and night. And what we see is that, in fact, they do experience sleep rebound, meaning that they need to make up that sleep. So these would be control fish in the white, and the gray is going to be things we kept up all night. And so we see increased sleep for those fish, including surface fish. By the time they get to the nighttime, so this would be during the day period of recovery, by the time they get to the nighttime, they're good. They've slept enough to make up for it. What I was also expecting is that we would see this dramatic shift in surface fish. Like, when you keep surface fish up, they're not going to be as resilient. They're going to have lots of stress turn on, but we don't see that. So I was thinking surface fish would have many genes that were upregulated or downregulated with sleep deprivation, and we would see kind of a moderate amount for cavefish, but in fact, we don't. So basically what we've learned from this experiment is that while they have less sleep duration need, they do still have their sleep homeostasis intact. So the amount of sleep they need, they still need it, and they still do experience sleep pressure. So then we wanted to know, okay, so putting aside sleep for a moment, how robust is the circadian clock in caves versus surface? And so we wanted to know there has been quite a bit of study on circadian rhythms and osteonics and other cavefish, but it kind of was all over the place as far as the results. We couldn't really make too much generalities about, do they actually have a pretty robust clock or not? And they do have, even though they don't have eyes, they do have a pineal gland that is very light sensitive and entrainable, it seems. And so what we did is we sampled cave and surface fish around the clock. And we did this for the 3 different caves I've been talking about throughout the talk, as well as surface fish. And we put them through RNA-seq. And we wanted to know how many of these were rhythmic. And so Katia Mack was actually the postdoc on this project. And so what she found, she did a remarkable job with this work, really cool results, and putting things together after not studying cavefish or circadian rhythms. And so she found that, generally speaking, surface fish have about 539 genes we would consider rhythmic, meaning that they're actually following some kind of pattern throughout the day, throughout a 24-hour period. 77% of those 539 genes appeared to be non-rhythmic in at least one cave population. Now, that's interesting, but what's remarkable is that 22% of the genes that were found to be rhythmic in Choi, in the surface population, were arrhythmic in all 3 cave populations. And what this looks like when we plot it on a heat map is that, so here's Choi, and we have things that are, 6 a.m., they're really, really cranking on, and then by the, late afternoon, evening, they're turning down, but by 2 a.m., they're cranking back again. Okay, so that's, you can kind of get a feel for the rhythmicity of those genes. When we look at the cave populations, they are all messed up and in different ways. And so this is convergent, it seems like it's convergent loss on these transcriptionally rhythmic genes. Their rhythmicity is convergently lost, I should say. Further, when we take what we think is the core kind of circadian genes, the core clock genes for zebrafish, and we say, okay, what do those look like as far as arrhythmic or not in cave populations? The darkest colors are arrhythmic in all 3 cave populations, and so and the light gray are 2 populations of caves that are arrhythmic, and super light gray would be arrhythmic in at least 1 population. And so we see parts of the core circadian clock seems to also be quite dysregulated. Now, we wanted to look at this and say, how much linked together is circadian versus sleep? And so we created CRISPments for ROCA A and also for A02, but I'm not going to show you that data here. But ROCA A is a key clock component. And in the surface mutants of CRISPments, and remember, CRISPments are going to be, they're not germline transmitting, they're more like mosaics, so it's not going to be perfect. But they do sleep quite a bit less. So when we mutate the ROCA A and make it basically not functional anymore, we can see that we actually see a decrease of nighttime sleep in these guys. And it seems to be significant. So this is work in collaboration with Joanna Qualco, who is the CRISPR lady for all of Cavefish. So, and James Yeager, who is actually a postdoc now at Stanford. So, okay. So the answers to the questions are, is circadian cycling intact in Cavefish? No, it does not look like it is. And the core clock genes really, some of them are seriously missing their proper expression. And it seems to be a convergent, different types of losses. And then finally, our circadian clock genes may be impacting sleep phenotypes. So I'm gonna stop there. I wanted to show you some cool new data, but I don't think we have time. And so I'm happy to take questions and go from there.
All right, thank you, Suzanne. If everyone would maybe unmute themselves or get their reaction button ready, give her a round of applause for that wonderful talk. Any questions? You can feel free to say them out or put them in the chat. Yeah, and I can just start with one. I was just curious about whether there's any kind of evidence for their kind of different like stages of diversification across these traits, like maybe morphological goes first and then behavior, and whether or not there's any convergence across different lakes in that order.
Yeah, that is a super cool question. And we're hoping that the Texas populations will start to, and that's really what I was hoping, is that we would bring them in the lab and be able to be like, oh, look, it's all behavior. Behavior comes first. But it seems like it's a mix. And then another postdoc in my lab, Danielle Drebeck, is she's actually looking, she's taking as many cavefish as we could get our hands on across like the fish tree of life, and saying, can I use phylogenies and mapping character states to try to understand that? So that's one of her goals. And so, yeah, it's a totally cool question, but we don't know. Yeah. And there's so we have a paper that's in revision at Current Biology right now that shows there's some genes that work pleiotropically for both, right? So OCA2, this particular paper, hopefully it will be accepted soon. It shows OCA2 influences albinism, but it also is necessary and sufficient for sleep phenotypes too, for at least part of the sleep phenotype. So that's kind of crazy, but it does make sense because it influences the catecholamines, and so the production and what goes where. So, it does make sense that it would be super pleiotropic.
Yeah. And Umi has a question of, where do the cavefish?
Yeah, they just kind of, they don't like settle on the bottom like a shark would, like and so they seem to just, I don't know. I guess they're not like laying on the bottom, but they are kind of down. So.
Hi, Suzanne. I have a question about the kind of shift in feeding behavior that you were seeing. Does that happen in the wild? So do they feed exclusively at the bottom, or?
Yeah, so we would love to see that. I think it's actually a stress behavior that got co-opted when they went into the cave for feeding. Because the Texas fish, when I brought them into the lab at first, they started to do that when they were introduced into new tanks. And so I think it's like a novel environment, exploratory stress type of thing. And then it was just like, oh, this is actually a really efficient way to get food too. And so we don't know. And actually before COVID, we had this we were supposed to leave March 23rd of last year to do 3 weeks of field work to actually get video of fish in the field. And so we'll get there. We'll get some quote, but that'll be awesome.
Do you see like any differences in jaw morphology across cave versus surface, or?
Yeah, and actually, I'm not the best person to talk to about that, but Josh Gross has done an enormous amount of work mapping this and also the asymmetry. And so, and I'm not exactly sure how much it affects the lower jaw, but I bet you it affects some of the upper jaw. And then the number of teeth are different for sure between cave and surface. But whenever the eye goes away, it actually causes a lot of collapsing and rearranging of the bones that you might not expect. And so, yeah, some of the head shape changes, and it's also the asymmetry. There's significant asymmetry on either side too. So
cool.
Yeah, that's actually a really good idea. Cynthia said to keep fish awake, you could just like do a jet of water bubbles instead of shaking, and that would be a much more a less jostling way that would be much better. So that's an excellent idea. We should do that. Thanks.
Easy time.
Yeah. All right.
What's going on with you were saying that there's like 5 populations that everyone usually focuses on, and there's like maybe 30 in total. What's going on with the other ones? Are they like not doing the same thing?
Yeah, so okay, so there's a couple populations that, like, whenever I said, so there's a couple populations that are hybrids that people don't mess with too much because of that. And so like, for instance, this one is definitely a hybrid, and that's why it's kind of a weird one that people work with. It does come up. There has been some work on it. Some of these are harder to get into physically, and then also some of these there were historical collections made and then taken into the lab before it was so hard to get fish out of Mexico, and now it's more difficult, and so making these collections and getting like lab stocks is a lot harder. So but there's also all kinds of fun and interesting cave systems. There's one that will have a paper coming out with Nick Rohner soon that's also a similar hybrid. Oh, I didn't talk about this. Is what I was going to talk about. Chica Cave, which we use a lot, is basically a hybrid cave. So like, this is a cave, this is a surface, and like these are samples from Chica, and you can see it's just a big old mess. It's like 75% cave, 25% surface, and Caballo was like that too. And that one's crazy because it has a sinkhole that shines light on part of the cave, and there's eyed cavefish in that part. And then there's, in the other part, the dark part of the cave, there's eyeless cavefish. And people who've been there say that the eyed cavefish actually chase the eyeless ones back into their corner, but I don't know what happens at night. I'm like, that doesn't hold up at night. They must be like interbreeding or mixing, but we see the same kind of pattern as I'm showing you here for eyed and eyeless cavefish in that cave too. So yeah, so there's all kinds of cool stuff, though some of them are harder to get to, and some of it's just logistics. So what was it? In cave surface fish crosses, do the offspring have eyes? The F1s is dominant, and in F2s you get the entire distribution like you would expect for a polygenic trait. Yeah.
All right. Okay. Maybe we'll let you go to see the speciation talk.
Oh yeah, that's right. Okay, we'll see you guys all there.
Yeah. Thank you so much for coming.
Bye. It was awesome.